like to briefly introduce you about Dr. B.R. Ambedkar Center for Biomedical Research and FBR. Dr. B.R. Ambedkar Center for Biomedical Research came into existence in March 1991 on the occasion of the birth centenary of Baba Saheb, Dr. B.R. Ambedkar. The mandate of the center is high quality postgraduate education and research in biomedical sciences. ACBR presents a unique combination of basic and applied research in the field of human health and diseases, which is first of its kind in the Indian university system. ACBR possesses a unique structure having an interdisciplinary goal-oriented approach with an object to meet the high standards of research and teaching in biomedical sciences. Specialized laboratories rather than conventional departments are the fundamental unit of ACBR. And this structure provides flexibility and enhances the opportunity for wide, you can say, intellectual interaction, as well as national and international collaborations. The faculty of ACBR and other departments of University of Delhi and neighboring institutes such as ICGEB in Mass, DIPAS, Patel Chest Institute, ICPO, AIMS, ICGEB, etc. They collaborate in offering a MSc PhD program of research in advanced fields mainly such as infectious disease, cancer, Alzheimer's disease, and diagnostic techniques, etc. In addition to the teaching, the ACBR arranges many research activities, seminars, and lectures by visiting scientists such as under SERP, that is Summer and Undergraduate Research Program, and other specific scientific programs such as one of them is Frontiers in Biomedical Research, that is FBR. ACBR has also been conducting many national and international conferences at Delhi University. And FBR is being conducted by ACBR almost every alternative years, which further leads to the interaction of our MSc and PhD students with eminent scientists, not only from India, but from worldwide, which makes a very good opportunity for all our MSc and PhD students to have interaction with eminent, you can say, scientists. So with these words, on behalf of ACBR and Delhi School of Public Health, I welcome Dr. Arun Sharma for today's lecture. So now I hand over to Dr. Meenakshi.
Thanks a lot, ma'am, for all the information about ACBR. So now I'll request Professor Madhu Chopra to introduce our speaker so that then we can officially start the session for today.
Thank you, Dr. Meenakshi. It gives me immense pleasure to introduce our today's speaker, none other than Dr. Arun Sharma. He is a senior information scientist at TBT Apex Biotechnology Information Center, abbreviated as BTIC, which is situated in International Center for Genetic Engineering Biotechnology, ICGEB we know, and is also associated with Translational Bioinformatics Group TBG at ICGEB. Dr. Arun, he completed his BSc degree from Kurukshetra University with botany, zoology, chemistry as major subjects. And then he completed his MSc in bioinformatics from CCS HAU, Hisar. And finally, he was awarded PhD degree in biological sciences by Academy of Scientific and Innovative Research, ACSIR. While he was working at CSIR headquarters, Delhi, as well as CSIR IMTech Chandigarh, and as far as I know, I think he was associated with Dr. Anshu Bharadwaj there. He is currently working at DBT Apex BTIC, ICGEB, as I told you, to support BTI's net centers, of which ACBR is also one of the part. And through his scientific as well as the computational skills which are unmatched, which he has. His research focus is on usage of artificial intelligence, computer vision, big data analytics, database developments, etc. Is well versed with almost all technologies, what I have told you, and I have been a listener to him earlier also. And he uses these technologies to solve important problems at healthcare, agriculture, social sectors. He has also worked into various research areas, including but not limited to development of computational resources for in silico discovery and design of therapeutic molecules, AI-based chest X-ray image classification, ML assisted prognostic biomarker discovery, where they use the clinical as well as the proteomics data and develop ML models to assist the prognostic development of the disease, and then development of QSAR models for prediction and design of novel inhibitors. He is also associated in one of the projects for prediction of major basmati paddy varieties using seed images, etc. So today he is going to talk about, the title is A Biologist View Towards the Utility of Artificial Intelligence Based Approaches to Combat COVID-19 Pandemic. So I hand over the stage to Dr. Arun Sharma to enlighten us with this technology, what he is going to talk about, how do we deal with COVID.
Thank you very much, ma'am, for such a kind introduction and inviting me at such a big platform to present our work. So I am very thankful to all of you for inviting me, and I hope I will be able to present my work in a very simple way so that everybody can understand what I did in past, and everybody will get a chance to learn the basic concepts of artificial intelligence and some other stuff that we did to tackle with COVID-19. So without delaying so much, I would like to start. I share the PPT. I hope the PPT is visible to all of you.
Yes, it is.
Okay, thank you. So the title of today's talk is A Biologist View Towards the Utility of Artificial Intelligence-Based Approaches to Combat COVID-19 Pandemic. So during the talk, first of all, I would like to introduce about the problem that we solved using the artificial intelligence-based approaches. And then I would like to tell something basics of the AI techniques so that everybody will come to understand how and what are the basic elements that are being used to perform any artificial intelligence based study. Then I would like to tell about you the application of artificial intelligence techniques to combat COVID-19, the global contribution and the contribution by our group. Then lastly, I would like to conclude the study that we performed. So as everybody know, today, according to WHO dashboard, SARS-CoV-2 has infected over 164.5 million people, and it is responsible for more than 3.4 million deaths globally. And currently available COVID-19 diagnosis methods, they are invasive, costly, time-consuming, and more out of reach of the rural population and resource-limited areas and hospitals. So the development of novel, non-invasive, low-cost, easily accessible, and rapid diagnostic methods is the need of hour in the current pandemic situation, where deep learning or artificial intelligence based methods, they are playing an important role in the classification of several types of biomedical images, including but not limited to cancer, COPD, diabetic retinopathy, and then chest X-ray and CT scan, etc. So let us start with the basics of AI techniques. So all of us, they regularly come to know or they listen some words like artificial intelligence, machine learning, and deep learning most commonly. So what are these? If we talk about the artificial intelligence, it is a broad area of computer science that makes machines seem like they have human intelligence. AI enables the machines to think that is without any human intervention, the machines, they will be able to take their own decisions. For example, robots are there, they can perform a number of tasks without getting instruction or decisions by their own. Then the second term, that is machine learning, it is a subset of as shown in the diagram, pink color. It uses statistical learning algorithms to build systems that have the ability to automatically learn and improve from the experiences without being explicitly programmed. So if we talk about the example of machine learning, a number of studies are there in bioinformatics where machine learning algorithms, they have been designed to for drug discovery, or they are being used for a number of tasks, biomarker identification, and then homology modeling, protein structure prediction, number of areas where you can see the application of these machine learning techniques. Then we comes to the deep learning. It is a machine learning technique that is inspired by the way of human brain filters information. It works the similar way the human brain works. It basically learns from the examples. It helps a computer model to filter the input data through the layers to predict and classify the information. So a number of studies you might be seeing nowadays that are using the deep learning techniques to solve a number of biological problems. Image classification is there and many other steps. So before going in depth of the artificial intelligence, let us learn some basic elements that are used in the back end of a Python script or an artificial intelligence algorithm that helps in training the models. And these models, they are being used for the classification or prediction of the unknown examples, right? So let us start with the basic elements. So first of all, we comes to the neural networks. A neural network is a network or circuit of neurons, or in a modern sense, an artificial neural network. It is composed of artificial neurons or nodes. So in the diagram, you can clearly see a simple neural network where input layer is there. You can see 3 layers there. First layer is input layer, then hidden layer, and then output layer. So what input layer does, it takes the input from the user, and then some processing is being done in the hidden layers, and then some output comes. If we talk about the prediction of gender, we can see height and weight. These are 2 input features that are being supplied to the these neurons, blue colored ones, and some weights and some biases, they are applied, and some calculation is done, and output is finally provided by the output layer, that is the gender of any person. So let us proceed further and go in some more depth. A neuron or artificial neuron is a building block of a neural network. It takes inputs, it performs some calculations and produce output, right? So there we can see there is a activation function. So what is the role of this activation function in any neuron? It is used to turn an unbound input into an output that has a nice predictable form. A commonly used activation function is the sigmoid function. That is example of a activation function. So if we go in some more details, if there are 3 things that are happening in this diagram. First of all, inputs x1 and x2, these are given, and these are multiplied by weight. X1 is multiplied by W1, then X2 by W2. Next, all weighted inputs, they are added together with a bias, that is P, shown by the green color. Then finally, the sum is passed through an activation function shown by the orange color box. And what this activation function does, as I already tell you, for example, a sigmoid, an activation function only outputs numbers in range of 0 to 1. We can think of it as compressing the minus finite to plus finite to between 0 and 1. So if I want to say in brief, big negative numbers, they becomes 0, and the big positive numbers, they become +1. So the process inputs forward to get this is known as key elements or words that are being that will be used later on, and these are commonly used during any machine learning study. If we talk about the back propagation, it is a process to determine which weight is better to modify a particular process. This process is called back propagation. So we repeat the process. We train our models, providing some input, and we repeatedly train models. And this repetition process, in which we adjust our weights to get the good results, this process is called back propagation. So during this process, we use some function, that is loss function. It tells about us about how good our prediction are, how good our models they are trained, right? So the better our predictions are, the lower our loss will be. While training the machine learning models, if the models they are performing good, then their loss value will should be very low. Or broadly, we can say better prediction is equal to lower loss. Or training a network, a neural network, we try to minimize the loss value, right? So what is a neural network architecture? In a neural network model, arrangement of different types of layers and their constituents is called neural network architecture. You have heard about a number of terms like neural network architectures, like ImageNet is there, ResNet is there, ResNet 50 is there, AlexNet is there. These are some popular neural network architectures that are being commonly used, right? The simplest type of neural network architecture is perceptron, where there is a layer of input layer and then output layer. There is a single hidden layer in the perceptron. So I directly comes to the images. If we talk about the image in terms of computer, how a computer sees an image. For a computer, an image is a matrix of pixel intensity values, which varies from 0 to 255 as shown below. Where the 0 value, it depicts the black one, and where 128 midway, it is used for the gray value, and 255 value shows that there is a white region in an image. So you can clearly see in these 3 images. First one is showing just zoomed image that is look like this, and these boxes within this, you can clearly see these are showing the pixel intensity values, and these pixel intensity values, they are varying according to the shape, right? The darker ones, they are having the lower pixel intensity values, and the white colored ones, they are having pixel intensity values near to 255. So what computer does, whenever we supply an image to computer, it learns from these pixel intensity values and it trains the neural networks, and in this way it try to identify any image, right? So this is the reason I am showing you, for a computer the image is a matrix, right? And this is I am showing you a black and white image, right? Here you can see just one channel is there, only 2-dimensional matrix you can see there, right? Rows and columns are there. Let us proceed further. I will tell you some commonly used terms that are used while image classification using the artificial intelligence techniques. CNN, you have heard this term earlier. If not, I tell you about what CNN is. In deep learning, a convolutional neural network, ConvNet, is a class of deep neural network most commonly applied to analyze visual imagery, right? If we perform any kind of image classification or video classification or the steps like those, we generally comes familiar with the CNNs, right? What these do, These involves the extraction of features of an image and convert it into lower dimension without losing its characteristics. If we supply to network, it's very large it to process. It can go memory of the computer. So what we do, as shown in this figure, our input is used by the computer or CNN in a CNN architecture, and image is used. Its different features, they are extracted from it step by step using these convolutional layers, and then finally these are merged together, that we say these are fully connected, and then we get an output. This is some broader overview of a CNN architecture. So if you want to understand in some details, we can see an image of size 224 by 224, and 3 is used for the in this slide. You can see we are having an image of 224 by 224. 224 is the height of this image, and again 224 it is the width. So first one is showing the width of the image 224, and its height is also 224, and 3 depicts that it is a colored image, right? In case of black and white image, it will be 1 instead of 3, right? So if we proceed without convolution, then we need to, if we multiply these, we get around more than 1 lakh numbers of neurons in input layer. But after applying convolutional layers, the input tensor dimension is reduced to 1:1, 1 multiplied by 1, and then lastly 1000, right? As shown in this diagram. Let us proceed further. Images are encoded into color channels. The image data is represented into each color intensity in a color channel at a given point, right? The most common one being RGB, which means red, green, and blue. As shown in this figure, you can see this is a colored image. It can be divided into 3 channels: red, green, and blue, right? Each channel contains the pixel intensity values for different colors. Red one contains the pixel intensity values for the red colored, then green one and blue one respectively. In this similar way, the information contained into colored images intensity of each channel color into the width and height of the image. The intensity of red channel at each point with width and height can be represented into a matrix. Like the same goes for the blue and green channels. So we end up with having 3 matrices, right? And when these combined, they form a tensor. So what is tensor? Tensor in simple terms, it is a generalization of factors and matrices, and is easily understood as a multi-dimensional array. We can say a tensor is a multi-dimensional array. Let me tell you something that is going on in these neural network architectures. While processing any image through the convolutional layer, we apply some filters to this image. And the filters, what they are? The filters, they are 2D matrices of numbers, right? That we show in the black and white image, right? We were having 2D matrices there. Similarly, these filters, they are 2D matrices having rows and columns and containing some values. In this diagram, the white color one, you can see a filter of size 3 by 3. There are 3 rows and 3 columns. It is a vertical Sobel filter. This filter is named as a vertical Sobel filter. You can learn about this more through the Google search. And these filters, they are applied in convolutional layers to reduce the image size and to transfer important pixel intensity values from one convolutional layer to the next layers, right? So dimensionality reduction is the task of these filters. They capture the important information and they transfer these information from one convolutional layer to the another without losing any information. The important features of an image, they are transferred from one layer to another layer. In this diagram, this filter is overlaid. If you see this gray and black colored image, this filter is overlaid on the image, and the image size is, you can see, 4 by 4, right? 4 columns and 4 rows. So let's proceed how this reduces the dimensions of an image. If we see, they are multiplying the image value or pixel intensity value with the filter value. If you remember from the previous slide, you can see the first box contains value 0 image, and the first one in the filter, it is -1. So in the next slide, you can see that 0 is multiplied by -1, we get the result 0. And similarly, 50 is multiplied by 0, and then 0 is multiplied 1. Then in the second row, each element is multiplied by each element of the image, right? So you can see in this slide, we get majority of values at 0. We can say these are meaningless for the computer, and you can see 62 and 33 values, right? Whatever values come as a result, what we do, we sum up these results, right? So if we sum up 62 and -33, we will get 29 as the value. And in the next slide, you can see our filtered image, it contains the first dimension, that is 29, right? Similarly, we proceed step by step, single step at a time, overlapping the image with the filter, and we get this reduced image that is containing the right one, containing 2 by 2 size. Means 2 rows are there and 2 columns are there, right? So if we talk about any formula, yes, there is a formula to calculate the image size after applying the filters. As shown in the bottom, you can see there is image size is n by n, and then if we get the dot product of this one, applying filter f by f, then the formula that can tell about the minimized image or filtered image, you can see n minus f plus 1 multiplied by n minus f plus 1. It will give us the size of the minimized image. Here n value is 4, and then 4 minus 3 plus 1, and then 4 minus 3 plus 1. So you can clearly see from here, that is 4 minus 3 plus 1. 4 minus 3, that is equal to 1, and we add 1, so we get the value 2, right? And similarly, after in the second one also, 4 minus 3 plus 1, and that is 4 minus 3 is 1 plus 1, we get the 2. So we get the size 2 by 2, that is shown in the right side, right? Containing 29, minus 192, minus 35 and minus 22 values. So this is the simple way to tell you that filters, they reduce the image size, and they play a very important role in the edge detection, right? As shown in the next slide, here is an example of image to which we applied vertical Sobel filter that I described in the previous slide. You can clearly see vertical edges, they are visible as in the right side image. You can see vertical lines or vertical structures, they are prominently seen in the black colored image, right? So these filters, they are very important to apply if you want to detect some structures in the edges of the images, right? So these filters, they can be applied for horizontal edge detection as well as vertical edge detection. In this slide here, horizontal Sobel filter is applied to detect the horizontal edges in the image. You can clearly see the difference between these two images, right? I proceed further. Then there are some commonly used terms. Stride is there. So what is stride? stride denotes how many steps we are moving in each steps in convolution. By default, its value is 1. So as shown in this figure, 3 by 3 filter is applied. 3 by 3 filter is of size 5 by. So on the right side, you can see feature gets. So we proceed one by one. Next, we will take it on the right side, taking one step. So stride denotes how many steps we are moving in each step by in convolution. So if we talk about padding, it is commonly used to keep the image size, or it is used to maintain the dimension of an output image. Padding, it is used to maintain the dimension of an output as in input, right? If we want that our input size remains same even after applying the filter, we use the padding. In this diagram on the left side, you can see the gray colored border boxes. These are the paddings. What is padding? It is a process of adding zeros to the input matrix symmetrically. In the following example, extra gray blocks denote the padding. It is used to make the dimension of the output same as the input, right? You can see in the left side, we are having our image with padding applied, and after applying the filter, 3 by 3 filter, green colored box is the filter applied. And after applying this, if we talk about the feature map, that is a reduced image, or we can say pattern from an image in a convolutional layer. Its size remains 5 by 5. Originally, the image was of the size, as you can see, sky blue color. Its original size was 5 by 5, and after applying the filter, the new image is containing the 5 by 5 size. So it prevents the reduction of the input. So without going in more details, let us start with some architecture or some layers in convolutional layer. You can see there are 5 different layers in any convolutional neural network. First layer is the input layer, starting from the left in the figure. Then there is convolutional layer, and that is accompanied with a activation function, that is ReLU function. Then pooling layer is there, fully connected layer is there, softmax is the 4th layer, and the output layer is the final and the 5th layer in any this convolutional neural network architecture. So I tell you the things one by one. So convolutional layer I have already told you. Then we comes to the pooling layer. What is a pooling layer? Pooling layer is used to reduce the spatial volume of input image after convolution, right? It is a process, in-depth process, that I am telling you how the convolutional layers, they reduce the image size. What is the concept behind that? If you see the left box, it represents our image, input image, and we applied a filter of 2 by 2 on this, shown by the pink color in the left box itself. So you can see in a pooling, here we have applied the max pooling. So what we do, we apply filter 2 by 2 and we get the maximum value from the input image. So you can see 1, 1, 5, 6, these are the 4 values under the filter. So maximum value is 6. So our reduced image, it is containing the 6 value. The right one, the right box, you can see 6 value is there. Then if we slide this filter further, we can see out of this 2, 4 and 7, 8, we get the maximum value as the 8. Similarly, if we talk about in the bottom, 3, 2 and 1, 2, these are the element values or pixel intensity values. Out of these, 3 is the maximum. So right side box, it contains the 3 values, and so on, right? So it is used between two convolutional layers. If we apply final convolution layer after convolution layer without applying pooling or max pooling, then it will be if we apply layer after convolution layer without pooling or max pooling, then it will be expensive, and we do not want to wait for the training of our models. So max pooling is the only way to reduce the spatial volume of the input image. In the above example, we have applied max pooling in single depth slice with stride of 2, right? Stepping of 2. So 2 stepping is there. You can observe 4 by 4 dimension input image is reduced to the 2 by 2 dimension. Let us proceed further. What is a flattening layer? It is used for converting the data into a one-dimensional array for inputting it to the next layer. We flatten the output of convolution layers to create a single long feature vector, and it is connected to the final classification model, which is called a fully connected layer, right? Fully connected layer. What is fully connected layer? It involves weights, biases, and neurons. It connects neurons in the one layer to the neurons in another layer, and it is used to classify images between different categories by training the models. Then the second last is the softmax or logistic layer. It is the last layer. It assigns the probabilities to different classes. Suppose we provide some unknown image to a model. After applying convolutional max pooling and these sort of layers, second last layer, it tells with which probability this unknown image belongs, right? This is the function of the softmax layer. In case of this multi-class classification, softmax function is used. Otherwise, we use the some other functions for the binary classification, right? The last layer, you know, outer layer, it contains the label, which is the form of one port encoded. Or simply we can say, for any unknown image, it assigns, this output layer assigns a class to any unknown image. So there are some more terms that are commonly used during the deep learning. Hyperparameter. What is a hyperparameter? Any constant parameter whose value is set before the learning process begins. The values of these parameters are derived via the learning or training the model. The example of some hyperparameter, there are a number of hidden layers, batch size, learning rate. These are some of the hyperparameters whose values we varies while training over the models to get the best prediction models. Then what is learning? It is the adaptation of network to better handle a task by considering sample observations. So learning involves adjusting the weights of the network to improve the accuracy of the result. This is done by minimizing the observed errors, right? Learning is complete when examining additional observation does not usually reduce the error rate, right? While learning, it is we can say we are training our model, or our model is learning. So what the model does during that learning, it reduces the error rate, right? So we use some hyperparameter, that is a learning rate. But is it the learning rate defines the size of the corrective steps that makes more the model takes to adjust the errors in the each observation. High learning rate shortens the training time, but a lower ultimate the accuracy. While a lower learning rate take longer time, but the potential for greater accuracy. So this learning rate value should be used in the increasing order. If you will use a learning rate value much greater or higher value, your learning of the model, it will be done very speedily, but the chances of getting accuracy are lower, right? So try this learning rate value in the increasing order. The default value for this one is the 1. You can start the value lesser than 1, 0.001, then 0.01, and then 0.1, like that. So there is the next term, epoch. What is epoch? Epoch can be defined as a process which in which the entire dataset is passed forward and backward through neural network only once. Since one epoch is too big to feed the computer at once, we divide it in several smaller batches, right? Uh, in data in forward manner through our neural network. Right? So if we do this process, supplying one image at a time, it will be very much time taking. So we supply our images in the batches, right? So it's the batch, what is it? It can define total number of training examples present in a single batch, right? This is the batch size. Suppose we are supplying 32 images at a time to the neural network. Our batch size will be the 32, right? Number of iteration is the number of batches needed to complete one epoch, right? So finally, we comes to some practical part, that is training data set. What it is, we are having our data of images, and we generally divide this data into three different parts. First one is the training data set used for training the models, right? It is the training data set. Then second one is validation data set. It is used for the validation of training models, and it helps in the selection of best performing models. Suppose we are training our models applying some images. Some portion of these images it will be used for the training of the model, and some portion it will be used for the validation of model, so that we can judge which type of hyperparameter they are providing us the better accuracy. And the test data set, it is used for the evaluation of best performing models, right? So the models that are the best model that are retrieved after the validation, using validation data set, we check those models again, how good they will work practically in real life. We use the test data set, right? I will tell these in the study that we have performed, right? So before doing that, I am telling you in very brief. Then we generally use the image augmentation. It is what it is. It is a process to modify the images. What type of modification? We can change the background of image. We can flip the image horizontally, vertically. We can rotate the image. We can sear or blur, or we can produce noise in the images. It helps in increasing the data set size, its diversity, and in the generalization of models, right? And then we comes to know a term, that is image processing. While training any image classification model, we pre-process our images. What we do in that, we resize our image, so that in practical life, sometimes if we do not capture the images in the standard conditions, then the images size, they may be varying. The images, they may be having different sizes. So in order to bring them into a similar size, we first of all resize the image. We normalize the image through the rescaling. We divided the pixel intensity values throughout image by 255. Then we assign the mode of images. For as I told you earlier, for colored images we can use the 3 value, right? These are some pre-processing steps we do before starting training our model, right? Or during the training of the model, the Python scripts, they can automatically do these sort of things. So designing of deep learning or AI model architecture, it is a very important process that includes adding the convolutional layers, pooling layers, dense layer, or dropout, fully connected layer, etc. So this is a flowchart diagram to show you, for designing any image classification algorithm, what we do. First of all, we will be having some images in JPEG or PNG format with us. So what we do, if the images, they are very less in number, we produce augmentation of these images. We augment these images, we rotate some images, we flip the images, we blur them, or crop them. These kind of modification they are done. That is known as image augmentation process. And after doing that, we will be having with us a data set ready for training the models. So what we do, we divide this data set into three parts: training, validation, and test data set. Training data validation, you can see it is used for the models. So training data set, what we do, we first of all pre-process our images. We resize, rescale our images, and we define the number of channels, one for gray color images or black and white images, and three for the colored ones. Then we apply different, we try with different convolutional layers containing different number of filters, filter sizes. Then we apply the pooling layer. So in any convolutional neural network, we can have multiple convolutional layers, right? And each convolutional layer, ideally, it should be followed by a pooling layer, as shown in the dotted blue color lines, as shown by this. And you are seeing here a new word, that is dropout. It is used to extract only the meaningful neurons while training the models. So dropout, what it does, during the training process, it activates certain number of neurons so that model overfitting can be stopped. So what is model overfitting? If our model learns from the examples that we are supplying to the model and it fails to predict the results with unknown cases, then we can say our model is the overfitted model, right? It only predicts good for the sample that we used for training and evaluation of a model. If it do not perform well on the real life examples, then we can say our model is the overfitted ones, right? I am trying to keep the words as simple as possible. Then after dropout, flattening layer is there, that I already have told you. Then finally fully connected layer and the output layer is there, right? So we get multiple AI models following this model training process. So how we get the best ones out of those? We calculate their accuracy using the validation test dataset, and the best models, they are used on the web servers or they are used on the apps, and where they are being utilized at the end users, they supply their images, and these prediction models, they give the prediction results, right? So I will quickly go through these slides without taking much time. I have already told you a number of things that are being used in these slides. These are some open source libraries that you can use for designing any image classification work or these convolutional neural network architectures. You can take help of these libraries. And also I have provided three very simple links that you can see some working examples to learn their uses. These libraries uses can be learned using the links given at the bottom of this slide. So you can try at your end. So if we talk about the application of artificial intelligence technique to combat COVID-19, if we talk about the global contribution, it is being widely used. Artificial intelligence techniques, they are being widely used to manage the crowd at hospitals and curtailing the rumors. They are used in the COVID-19 drug discovery, in designing the inhibitors against SARS-CoV-2, and it is being widely used for the surveillance or to detect the outbreaks through the AI techniques. And it is also being used in the prognosis to detect what are the chances of a patient that is admitted in the ICU, how many chances are there the patient will survive or not. Then early diagnosis, if somebody wants to have some idea whether he or she has been infected with SARS-CoV-2 or not, in that area is also chest X-ray and CT images, they are being used. Then I comes to the usage of artificial intelligence in COVID-19 diagnosis. So in this slide, you can see we are worked in prognosis area, the bold ones, and early diagnosis and drug discovery. I am happy to tell you that yesterday our article related to COVID-19 patient prognosis using clinical and proteomics feature has been published in Frontiers Genetics. Use the clinical and proteomic features to survival chances of a COVID-19 patient. So in today's talk, I will discuss the uses of AI in COVID-19 diagnosis. Studies have shown potential of deep learning or artificial intelligence-based algorithms in the classification of COVID-19, non-COVID-19 CT images. So CT images, they can be used for the classification into COVID-19 non-COVID-19 images. Attempts have been made to classify COVID-19 non-COVID-19 chest X-ray images. However, CT images are costlier to generate. Even the facilities available at limited hospitals. Currently, if we talk about the limitations of these available techniques, so currently available chest X-ray images-based COVID-19 classification algorithms poses several limitations in data pre-processing and lack of systematic approaches, such as they use the wrong age group selection criteria to classify different types of normal disease or chest X-ray images. They use limited augmentation types. They use the commercial software for AI-based models training and testing. These are some limitations that we observed while designing our experiment. So these were the limitations of the studies existing that time. So if we talk about the advantages of AI techniques in chest X-ray images-based COVID-19 diagnosis, chest X-ray images, they are easy to obtain in majority of hospitals. Use of chest X-ray images-based diagnosis is a non-invasive method, right? Multiple infections such as TB, pneumonia, SARS, etc. can be considered simultaneously while the classification of chest X-ray images. And rapid diagnosis, even in high disease burden areas, they are very rapid in their action. In high disease burden areas, you can supply lakhs of images, chest X-ray images, to find out which ones they are COVID positive or not. They are easy to use and they are scalable. They are highly effective in preventing the spread of the disease. If somebody is having the COVID infection, he gets or she gets the chest X-ray done, they can submit their chest X-ray images on any app or web server and come to know whether he or she is infected with COVID-19, SARS-CoV-2 or not, right? So in this way, if the prediction result is COVID positive, then the person, they can isolate themselves, right? So we comes to the main point, application of AI applied by us to combat the COVID-19. So what we did, we used the artificial intelligence-based approaches to classify chest X-ray images into COVID-19 and other infectious diseases, right? So this slide shows the basic study flow. What we did, we collected the 5 different types of, we can say 5 different class of chest X-ray images from publicly available data sets. The normal images were there, COVID-19, non-COVID-19, pneumonia and TB images were there, right? What we did, we performed some augmentations on these images because we designed this study in the April 2019, April 2020, right? That time the chest X-ray image dataset was very low. So to increase the dataset size, we augmented our images. We applied number of different type of augmentation that I will show in subsequent slides. Then we divide our dataset into training and external validation dataset. The training dataset, it was again divided into 90% as the training dataset and 10% as the internal validation dataset. Using the training dataset, the AI-based model, they were trained, and this internal validation dataset was used to get the best, to know which models they are best ones. And finally, the best models, they were further evaluated using the external validation datasets. These are internally hosted on a web server that we have not shared the link knowingly because it is a research server only. And these models, they are being used in the backend to predict the COVID-19 status of any patient using its chest X-ray image as input. So this slide shows the number of images in our original data set. You can see less number of images were there, and that is what is techniques to increase the data set size. So here you can see a total of 350 corresponding to 5 different classes we were having. We divided these into 90% as the training set images, 317 total in number, and 10% as the internal validation data set 1, external data set 1, that is 35 images. And after training and developing the models, some data was accumulated or it was collected from the literature between March 19, 22 and May 29th, 2020. 75 images of COVID-19 positive patient were collected to evaluate our prediction models. So this slide shows a total of 25 different types of augmentation techniques applied by us. Original image shown at leftmost position, and these are different augmentations that are applied to this original image. So we sharpen the image, we make it blue color, or we applied a number of different type of rotations, 60%, 90 degree, then 120 degree, 140 degree. You can see from this slide, we also cropped the images. In this way, we increased our size of the data set. So broadly, you can see we were having initially 603 images from the publicly available resources. We applied some selection criteria. We used only those images that belong to patients having age greater than 18 years, and then chest X image with PA view, posterior anterior view only, they were used for training the these artificial intelligence based models. So we were having 352 images, and a different type of data set used for AI based modeling. We created 27 different types of data sets using applying augmentation techniques. So in that set 1, it was consist of original images, you can see, and data set number 2 to 26, it was containing a single type of augmented images at a time. Suppose data set 1, it was containing rotated image through 60 degree angle. Data set 3, it was containing 120 degree, and so on. And the last data set number 27, it was containing all the augmented images plus original images, right? So it was containing 9,152 images. If you want to see further details, these are given in the supplementary materials. So I proceed further. These data set, 27 different type of data set, they were used for training the models using the different hyperparameters that are shown in this slide. So what we did, we used the batch size as 16 for 26 different models that were trained on the lesser number of images, single augmentation at a time, and original images based model A showing the original images, or single augmentation based models that use the hyperparameters that are shown in the section A. And the combined model that was containing original and all augmented images, the hyperparameters that were varied apart from the section A, these are shown in the section B, right? Only number of iterations and batch size and number of epochs, they were varied for the larger data set. That was the data set number 27, right? So here this shows the flow chart of our study. We used 27 different data sets for training and evaluation of the models. You can see I would not repeat the things that I have told earlier. We were having 29 different models corresponding to 27 different data sets. The data set number 27, it was used with additional number of epochs. So we are having 29 models for the 27 using 27 different data sets. So applying different strategies and applying some filters, that was after training the models, we apply some filters to get the best models. What were those? Highest accuracy on the test set for all 5 classes data set. These type of models, they were considered that are I will in the subsequent slides. So if we talk about the performance of models, or these 3 different models, from table 3 you can see these are 2 different models. One trained and evaluated using the rotate 120-degree images, and the second model, it was trained and evaluated using 140-degree images. What was the reason of taking these 2 models? If you see through these red color circles, some of a model that is 120-degree, it was performing very poor on the normal COVID-19 and new COVID-19 images, while model trained on 140-degree images, it was performing good for these. And you can see rotate 120-degree, it was performing very good for the prediction of non-COVID images and pneumonia and tuberculosis images. So these 2 models, they collectively perform very good for all 5 different classes of images, right? So table number 4, it shows us the performance of the combined images based models that were trained with 24, 49 and 101 approach, right? So you can clearly see these models, when they were these combined images based model, when they tested with combined images containing all type of augmented images, they did not show any good performance. So what we did, you can see from this slide, we checked the performance of these combined models using one single type of augmentation at a time, and using the rotated 60-degree images, we got the best performance of these 3 models. Out of those, model number 3, that is 101 approach based, it showed good performance for 4 different classes of images, right? Except for the new COVID-19 images. So because it performed good for 4 classes, we also used this as a, we also shortlisted this model, this 101 approach based model, as our the best model. So in conclusion, all 3 best performing models, that is rotate 120-degree, rotate 140-degree, and combined 101 approach based model, along with the Python scripts used to train and validate the models, they have been provided freely for the public use at the link given there, right? So if we talk about the performance of these models in detail, we can see the 120-degree images based model, it wrongly predicted the normal images as the tuberculosis images. Diagonal values, they should be higher ones. Suppose if we see normal is normal, predicted as normal 14.29. I have already shown you in the previous tables. It performed good for the non-COVID pneumonia and tuberculosis images, showing the 100% accuracy in their prediction, and performed very poor for the new COVID-19 images, and normal images. If we talk about the 140-degree rotated model, it performed good for the upper 3 ones, that is normal images, 100% performance, 100% accuracy, then COVID-19 100%, and for new COVID-19 that was containing 75 COVID images, chest X-ray images, it showed 94.64% accuracy. It was quite good. This is the performance of the 101 approach-based combined model. It performed good except for all the 4 classes except the new COVID-19 class, right? and these results they are for the using the 60% degree rotated images as the external evaluation images, right? And this is a screenshot of the web server that we use internally at our end to predict the COVID-19 status of a patient using its chest X-ray images, right? This is the prediction phase, screenshot of the prediction here we have provided the option to the users to submit their images, and we ask whether the person want to contribute that image for the future development of the COVID pred server. We named this prediction server as the COVID pred server. So there anybody can upload the chest X-ray image and push the classify button, and these sort of results are retrieved. What these are showing, it is showing that the submitted chest X-ray image is predicted as COVID-19 case by all three models that I described in previous slides, and it is showing the probabilities values in the descending order. As you can see, the different probability values to belong to different classes. And finally, I would like to conclude that AI-based classification can help in rapid diagnosis of COVID-19 and other major infectious diseases. The models developed by us are a proof of concept that cost-effective, user-friendly, non-invasive AI-based methods can be developed for COVID-19. AI-based models developed by us may be evaluated for its use in clinics as diagnostics or clinical management of the patients. In future, with the availability of more and more images representing diverse cases, the efficiency of the models may also be scaled up. So lastly, I would like to thank all the persons that were behind me. First of all, I would like to thank our director, Dr. Dinkar M. Salunke, for providing the necessary infrastructure and all the facilities to conduct this study. Then I would like to thank Dr. Dinesh Gupta, our mentor, our group leader. He was always there to help us and guide us through different phases of the study. His guidance has been very useful for us. And lastly, I would like to thank Siba Rani, that was a contributing author. She contributed equally to this work. She was always there to help me for data collection, model training, and all those sort of stuffs. And thank you very much.
Thank you, Dr. Arun. So it was really a nice talk. You first gave all the basics and then you told about your study. I can see Madhu ma'am has got some questions. She has raised her hand. So I'll invite Madhu ma'am to ask a question. Then we can go for the students' questions.
Thank you, Dr. Arun. That was a very lucid talk. You tried to cover so many basics for the students, even though these students are mostly from biology background. But it is highly commendable work because it is the first work I'm listening, which is using the X-ray images. And this has to be very useful for the smaller towns, and even in Delhi, we were not getting the CT scan results. 3 to 4 days we were getting after those, and till that time the disease progresses so fast. So my first question is, when you are taking these X-ray images, did you consider which day of these disease progress X-rays were taken? Because in the earlier stages, when it is a very small opacities, which is a glassy-like opacity, which is termed in CT scans, it is very low and it is undetectable in the X-rays and X-rays of the patient. So did you take into consideration which day the X-ray was considered or was taken by the patient, or water sample you have collected? or the disease has already progressed and you have taken those X-ray images?
Actually, we were having very limited images, so these were mixed type of images. The day information was not there, so these were random images, you can say.
So I think in the future development models, you might be requiring the day 5, day 6, day 7, those X-ray images correlated to the CT values of CT scans, whether the disease has progressed to a milder, to moderate, to the severe category. And that will be really useful for the user, because then we will not need the CT scan to be done. X-ray should be able to. So I think you will be modifying your model according to the day of the second question was same what you have already answered. You did not have too many images. So I was also wondering, mostly the AI based method required thousands of images or maybe hundreds of images to develop, and you have used whatever little data was available in March, April, May 2020. So are you looking forward to include more number of images and then redevelop your model and modify your model?
Yes, ma'am. I agree with both your suggestions, and thank you very much for these nice suggestions. First of all, definitely the stages information, if we keep with these images, it will be very useful while developing the new classification models, and it will help us in training the models in a better way. In future, we are planning to retrain our models using the larger data set, because that time we were having very limited number of images. But as we all know, now we are having sufficient data, and the data is publicly available too. So we will make use of that data, and we will try to develop some better models with the larger data set.
Another very curious question was, why do you think while rotating the images, the accuracy was improving? It's just a layman kind of thing. What was happening? You must have played with so many images. So why do you think it is behaving like that, or it behaved like that? And then while you were developing the training when you trained your model and then you predicted or validated your model, this was this rotation, like 60-degree rotation in training the model as well as 60-degree rotation in validation, these two rotations were same, or you were varying the rotation when you were validating it?
Actually, this is a very nice question. If you remember from the basics, we can see for any image we applied different filters, right? So when the filter is applied on a rotated image, in contrast to an original image, it try to capture, or it try to focus on certain areas, right? So while rotating these images, it try to capture certain regions that are being differently affected in different kind of diseases. So that is why I think this rotation technique worked in our case.
Okay.
And we were having very shallow network. We were having just 3 convolutional layers in our network. So we knowingly we avoided to use a larger number of convolutional layers, because for larger data set they might work good. So I think it's clear to you how this rotation it worked.
Yes. Now it is clear. Thank you so much, Dr. Arun.
Yeah, following the same question.
Aparna, may I ask a question?
Yes, sir. Please go ahead.
Yeah, so this is in relation to what Dr. Madhu asked. To improve the specificity of your models, did you look at any non-infectious X-ray samples like the ILD, interstitial lung diseases, which happens as a form of an autoimmune disorder, and that will probably also tell you some kind of, or give you a handle to look into as negative controls to get rid of the noise versus the like the test sample, so that later on when you get more and more access to more and more images of COVID-19, you can actually better filter and more train your models. That's one. The second one is that now that like post-COVID infections, mucormycosis is also coming up. That's also affecting the lung. So are you able to pick up any CT scans of this and then relate it to the COVID images to see whether any predictive models can be made? Whereas if an image pattern is coming like this, will that person be more susceptible to mucormycosis if at all he or she contacts? These are the two questions I had in mind. But very nice talk, just if I may add.
It will help us in improving our study. We will incorporate training models and we to get data containing this sort of problems that you have discussed.
I have one question, Dr. Arun. I'm Dr. Aparna from ICGEB. So what was the source
Yes,
of these images? Where did you get the images from? And
I would like to, yes, I would like to show you at the end of my presentation.
okay.
I do not know whether the PPT will be shared with all of you or not. Supplementary slides, they contain this information.
Okay, because that is the most important thing. The source for the images, the X-ray,
these are given in the, yes, these are given the links from where we downloaded these images. They are given in the supplementary material along with our published article.
Oh, so
you
this
can
is
get
not
the
from
links
any
from
hospital.
there.
So this is not from any hospital. This is from the database. The X-ray images.
No, actually, these were captured from different sources. NIH database of test X-ray images was used to collect these images, some of these images. Then some Cohen et al. Data was used. You can find the link in the published material.
Yeah, because I understood, because in general, whenever such systems are developed, a radiologist, since this is replacing a radiologist, X-ray based artificial intelligence is replacing a radiologist. So the validation is done by a radiologist, isn't it? Of course, if the data is available.
No. Actually, these are not the replacement of radiologists. These are the to assist them. Actually, if we are having a very huge patient load, and some of the images they are being filtered using these techniques, it will help in reducing the load of radiologists.
Yeah,
if we are having thousands of cases per day, lakhs of cases, then we can submit on web server or using these scripts, and we can come to know out of 1 lakh how many they are probably COVID positive or not. This sort of work, these algorithms, they can help.
Okay. Because I had this in my mind that there has to be a clinician to validate it, at least for the imaging data, the radiologist. But maybe it is.
There are studies that show that these AI algorithms, they have beaten the radiologist even. They are getting best results even better than the radiologist, expert radiologist. A number of studies you can find in the literature.
Till now, they have used those images which are well validated, already diagnosed people to build their model. In future, when the server is used for any layman, any person, any doctor, any medical person, then I think it is to be revalidated with the parallel diagnosis. But yes, for a screening kind of thing, like when he's saying thousands of images come, and then a fast screening is required, how many of them are like simple pneumonia, how many of them are tuberculosis, how many of them then it will be very really useful initially, and an enthusiastic patient himself wants to know whether he has COVID or not, and then he can validate it what stage it is. That is what I brought the question of staging. Like when you get this kind dense image, this is lesser denser or higher dense. So what stage I am, because getting the CT scan done is very tough and getting X-ray done is a little easier in almost all settings. So that way it will be very helpful in screening kind of thing. It cannot replace the radiologist. Ultimately, the thing has to go to the doctor for the thing treatment also.
Basically,
Actually, I would like to share with my own experience. I was having my cousin brother, and he got infected with the COVID-19, sorry, SARS-CoV-2, and he got his RT-PCR test done, and the report was normal. He asked me to tell he was having symptoms of COVID-19, but not getting detected using RT-PCR. So what I suggested to him, please send your chest X-ray image to me, and I will take a try on the web server that we have developed. So he did the same, and we uploaded the chest X-ray image on the web server. And two out of these three models, they predicted him as COVID positive. So what we said, I suggested him predicted as COVID positive using this web server. Please take care and isolate yourself from other persons in the family. He did the same, and after I think 4 to 5 days, the symptoms they got severe, and he again asked me what to do. I suggested him immediately go and meet to the doctor again and show that you are getting some more problem. He was facing problem in breathing also. So when he again got his test done, he was found COVID positive. So in this way, our web server may be useful for early diagnosis or early identification of the patient, so that uh, they can prevent the spread of disease. In this sense, this can be useful because the city facility is not available in each and every hospital. Very limited hospitals provide these kind of facilities, and but this chest X-ray machines, they are readily available, I think, at majority of these PHCs. So in that
But will
sense,
it not
it
also depend
can
on
be
the
useful.
quality of the X-ray?
Yes.
Yes, it
Because
depends
in
on that.
rural areas, you may have like outdated machines with not very good resolution, like the digital versus the film-based, and then the scan, how the way you scan it. Will all that not
Yeah,
factor into these things?
Yes.
actually, if I tell about, if you see our example, the person took image in hand and took photograph from the mobile phone only. So the resolution it was varying there, but the model they performed
That
good.
is very nice. And was it a film X-ray or a digital print?
Actually, it was film X-ray.
Oh, then it is okay. Then that is a good model then, because the film X-rays are not all that high resolution, and then depending on the camera of your photo, the camera or the phone or something, it may further reduce. But even then, your model could predict it. That's what you said.
Actually, images they were from different sources, so they were having different resolution.
Yeah,
So
no,
from the very beginning, the model was trained to learn these differences, resolution differences, because of the different sources of the images.
yeah, I'm praising your model that it is able to filter out all these minor
Thank you, sir.
differences. Nice,
Thank you,
very
sir.
nice.
Thank you so much.
Yeah. Dr. Arun, I will be interested in your other paper, which was related to your clinical proteomics
Yeah, sure, ma'am. We will share that.
data in Frontiers, because
Frontiers in Genetics, you can see the paper is published yesterday only.
okay, so I'll
So
contact you for the, if it is freely available, I'll get it.
yeah,
Otherwise,
it is freely available. It
okay.
is freely available from the Frontiers in Genetics journal website.
So it was really an interesting talk, Dr. Arun. Any other questions?
Yes, ma'am, I have.
Yeah, please go ahead.
Yeah, hello, sir. Myself, Tatak Rana, and your talk was quite nice. So my question is, initially when you told us about this image analysis of Abraham Lincoln, that pic, so in that you told us that the model is taking the specific pixels, specific characteristic thing from the image, not total image. It is taking the specific thing from the image into its account in order to predict. And similarly, CNN also is reducing the total database into small database. But in augmentation thing, what you have told, it is utilizing the spatial distribution of the infection. So if
Yeah.
the model has to, if the model has to consider the spatial distribution, how can it, consider the very specific thing from the image? It has to take all the database. So how can it reduce the database? No, matlab, how these two contrary things can work together? Because आपको पूरा 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 एक से आया है, उसमें इन्फेक्शन हुआ है, अगर मॉडल सिर्फ क्या कहते हैं सिर्फ वाइट पैचेस को मान लो एनालाइज कर रहा है, प्रेडिक्ट कर रहा है, ओके द कोविड इज देयर एंड पर स्पेशल डिस्ट्रीब्यूशन के लिए उसे बैकग्राउंड इमेज भी चाहिए होगा ना, वो भी तो डेटा पिक्सेल्स का उसे अकाउंट में लेना होगा। तो ये दोनों चीजें काम मतलब साइमल्टेनियस काम कैसे कर रही हैं?
आपकी वॉइस ब्रेक हो गई बीच में। Two things. What are the two things? First one that I have understood, you are telling about the background of the image.
Yeah. Okay. So my question was, if your model is also considering the spatial distribution of the infection, so it has to take in the account of the whole database of background also and of the characteristic part of the image. But in the initial part of the slide, it is only taking considering the specific part of the images, not whole images, in order to reduce the database.
Yeah, I got your question now. So I told you that the whole image, it is translated into a reduced image applying some filters, right? If you remember from the basics. So using this, what we do, we consider the only distinguishing features from the images. Taking the different class images, we try to identify which are the distinguished features that discriminate normal images from the COVID-19 or TB images and all different class images. So only the distinguishing features, they are taken on the next neural network layers.
हाँ सर, एक्चुअली यही था कि अगर वो डिस्टिंग्विश इमेज मतलब ले रहा है तो उसे बैकग्राउंड भी तो कंसिडर करना पड़ेगा ना इमेज का, तभी तो वो डिस्क्रिमिनेट कर पाएगा भी टीबी है, कोविड है, बिकॉज उनका डिस्ट्रीब्यूशन इन्फेक्शन का अलग अलग होगा, मतलब सेम नहीं हो सकता, तो उसे बैकग्राउंड भी लेना पड़ेगा।
It considers the both the features as well as the background. If you talk about the background, what it is the pixel intensity values.
Okay.
Nothing more than that.
And just a second question, whether your model can predict that if the person having both infection, suppose white fungal infection, which is is on upcoming thing, and COVID also, so दोनों में white पैचेज होंगे, so
actually these models, they are trained earlier. That time this type of data set was not available. We will try to develop some new models that will be able to work on these type of data. These models, okay. I am not sure whether they will work or not.
Okay. And just small question last, if
yeah, sure, no problem.
So in your project, most of the members, I am presuming that most of the members must become computer science background, bioinformaticians, computational biologists, and the clinicians also. But what about the people from the basic basic biological sciences? How can they contribute in the project? Because I'm from biomedical sciences. Suppose I want to be the part of the project. Suppose in future. So how the person from the basic biomedical sciences, biological sciences can contribute in this such kind of amazing project?
Anybody who has interest in programming, like me, I was a biologist only. If you see my background, I was having BSc with botany, zoology, and chemistry. Nothing computer world was there. And after that, I did MSc with bioinformatics, right? So that time only I came to know some programming languages are there. I am from a rural background and I am speaking from my village only. but I got interested in learning the programming languages. So because of those only, today I am able to teach you about these fundamental things about these neural networks or machine learning. Anybody who has willing to learn can learn by their own. Lots of resources are there. I have provided some links there. There you will get some sample codes. And before starting that, you have to learn the Python programming language. That is very simple to learn. So there is nothing like that. Everybody learn by doing the things. That I believe.
Okay, okay. Thank you so much, sir.
You are welcome.
Any more questions? Okay. And then thanks. It was a pleasure to listen to your talk. It was excellent, and I'm really glad that I heard it. And I look forward to take you as a speaker in our future seminars or webinars. Webinars are definitely better. We have more audience, and I'm really thankful for your. It was a very good lecture with basics. And people like me, I was correlating it with the visual pathways, because I could correlate the way you were talking about the neural pathways, and I teach neurobiology and physiology. So it was clearly how our brain basically filters, like the LGN, lateral geniculate nucleus. It filters the images, and then the visual cortex. We have different cells. So same way your neural input and output was there. So it is. I could relate it that way, and it is indeed very good in all the webinars. And this was especially very good for our students. They're very excited about Friday webinars, because then they will have they will know which area to go for in the research in future. So I'm really thankful to you for a wonderful talk, and looking forward to sharing in the future also. Thank you, everyone.
Thank you, ma'am. Thank you very much for compliment.
Thank you, Dr. Arun. Thank you. Bye.
Thank you, ma'am. Bye bye.
Okay.